What's going on, my YouTube family, as well as my pre-healthcare professionals alike? I hope that y'all are having a wonderful day. We're going to continue on with the science review portion of the ATI TEAS and start discussing the cell. If you haven't done so already, make sure you hit the subscribe button down below, and better yet, hit that bell notification. It lets you know when I post new content here on YouTube. And give this video a big thumbs up, that way other people that are looking for the ATI TEAS review videos know that this is a good video to help you pass it like a boss. Questions related to the cell test your understanding of the differences between prokaryotes and eukaryotes, as well as the various parts of the cell. You may be tested on your understanding of cell reproduction, including the processes of mitosis and meiosis. Let's get started understanding how the cell is important on the ATI TEAS. To begin, we have to understand cells in life. Life takes many different forms, from the simplicity of bacteria to the complexity of primates. According to cell theory, all living things are comprised of cells. Complex life forms have more cells and more complexity to their cell structures. Cell theory goes on to state that cells are the unit of the function of organisms. They are responsible for life functions such as digestion, circulation, reproduction, and immunity. The life cycle depends on two different types of organisms, autotrophs and heterotrophs. Autotrophs come from the Greek language and means self-feeder. Autotrophs produce glucose through photosynthesis and feeds themselves and other living beings. They are mainly plants. Heterotrophs get their nutrition from other sources. The prefix hetero means different. Animals eat plants and other animals to survive. The cell structures of autotrophs and heterotrophs are different. There are two basic types of cells that form the building blocks of organisms: prokaryote and eukaryote cells. Prokaryote cells are simpler, having no nucleus and lacking some of the complex organelles of eukaryotes. Their DNA is not tightly contained as it is with a eukaryote nucleus. Prokaryote cells are represented in two types of organisms, bacteria and archaea. Most organisms in these two groups are just a living cell with a flagellum for movement. A flagellum is also known as the tail. They replicate themselves through a process called binary fusion, in which they split apart, creating two exact copies of the same cell. Eukaryote cells are present in almost all of the abundance of life visible to the eye, from plants and animals to fungi and even some bacteria. They have membrane-covered organelles, including a nucleus that holds the cell's DNA, and they reproduce through either mitosis or meiosis. To begin, we have to start understanding the parts of the cell. A typical animal cell is filled with cytoplasm within the cell membrane. The cell membrane allows select substances such as proteins, enzymes, and chemicals to pass through while keeping others out. Resting in the cytoplasm are various organelles. Organelles serve to regulate the metabolic functions for the cell. The nucleus is the control center of the cell and contains the nucleus. The cell's DNA is contained in the nucleus, and it delivers information to control the metabolic functions of the cell. The nucleus produces ribosomes. Ribosomes are found throughout the cell and are synthesized proteins. The mitochondrion is the energy center of the cell, where glucose and oxygen are broken down into water and carbon dioxide. As a result of breaking these chemical bonds, energy in the form of adenosine triphosphate, ATP, is produced. The endoplasmic reticulum of a cell is the membrane where proteins and the building blocks of cellular life are built and stored. Rough endoplasmic reticulum has ribosomes attached, whereas smooth endoplasmic reticulum has none. Working with the Golgi complex or the Golgi apparatus, the endoplasmic reticulum assembles proteins and makes structures with those proteins. Centrioles are organelles that assist in cell reproduction, either mitosis or meiosis. Lysosomes capture the products of cellular function that the cell cannot use. They break it down into cellular waste. Plant cells are similar to animal cells in most respects. They have the same organelles, but they also have a cell wall that contains chloroplasts. Chloroplasts are organelles that aid in photosynthesis, through which plants use water, carbon dioxide, and the sun's energy to create glucose and oxygen. In understanding cell reproduction, we know that cells proliferate in one of two ways, through mitosis or meiosis. Mitosis is the way that the cells proliferate through sexual reproduction. In mitosis, cells produce an exact copy of themselves. Meiosis is how cells reproduce through sexual reproduction as well, but in this case, each daughter cell has half of the DNA of the original cell. In sexual reproduction, the daughter cells combine with each cell from another individual to form offspring, leading to genetic variations. Mitosis occurs when a cell duplicates itself. This can happen in single-celled organisms such as protozoa and bacteria and is how they reproduce. It also happens in other living organisms when they grow and heal. New cells are created with the same DNA as the original cells. When more cells are needed or a sexual stimulus is introduced, chromosomes and centrioles are also replicated. To begin the stages of mitosis and cell division, we know that prophase is the first phase of mitosis. In this phase, the nuclear membrane dissolves, allowing the doubled chromosomes to flow freely. Spindle fibers congregate around structures known as centrosomes to produce a spindle apparatus, which separates the floating DNA into separate poles. In metaphase comes an orientation of the spindle apparatus drawn by the centrosomes to push the DNA to opposite ends of the cell. During anaphase, spindle fibers retract, again influenced by centrosomes, pulling apart chromosomes into their V-shaped halves. Telophase ushers in a reversal of previous processes, with spindle fibers dissolving and nuclear membranes forming around the new chromosome pairs. At this point, mitotic division is all but complete. After telophase, the two daughter cells undergo cytokinesis. This is a single process in which the two nuclei are divided by cell membranes. There are now two identical twin cells with the same DNA ready for interphase. Meiosis is a more complex process than mitosis. Cells in meiosis go through two rounds of prophase, metaphase, anaphase, and telophase. These two stages are called meiosis 1 and meiosis 2. In prophase 1, it is similar to mitotic prophase in that the nuclear membrane disappears, allowing chromosomes from each parent to mingle. In this case, chromosomes performing a crossover in which similar chromosomes from each parent bundle together. An allele from one parent may replace an allele from another, causing genetic variation. In metaphase 1, anaphase 1, and telophase 1, they mimic the mitotic counterparts. The new chromosomal pairs, called tetrads, migrate and cytokinesis begins developing two diploid cells, containing a full but unique complement of mixed DNA, 46 chromosomes in humans. These two daughter cells then begin the process of meiosis 2. Prophase 2, metaphase 2, anaphase 2, and telophase 2 mirror the previous process. Centrosomes and spindle fibers push apart chromosomes. When the spindle fibers retract, chromosomes are pulled apart. When telophase 2 begins, each daughter nucleus has only one of each pair of chromosomes, 23 chromosomes rather than 46 in humans. This results in 4 haploid cells or gametes. I hope that this video has helped for you to pass your ATI TEAS like a boss the first time. If you haven't done so already, I want to invite you over to my website at www.nursecheung.com. There's additional resources for you to help you pass this exam. We've got practice questions as well as PDFs of the PowerPoints that I show in these videos. If you haven't done so, make sure that you follow me on my social media. I am on Facebook and Instagram. And until next time, I hope that y'all are having a wonderful day, and I will see you all again soon. Bye!